It's a slightly sticky afternoon in Dundee, so I wish you fresher weather, although it's really remarkably nice in Scotland at the moment. So I assume everybody can see the startup screen. I'll show you the systems check screen just to make sure everything is going.
Everything looks good.
Wonderful. So, Jalview. I'm sure that many of you are aware of Jalview in some way, shape or form. Its name comes from the fact it's an alignment viewer. Of course, it doesn't always, it's not just for viewing, it also edits alignments and copes with other kinds of data as well. So it allows you to annotate multiple alignments, create trees and view trees and structures associated with alignments. And the J, of course, is due to the fact it's written in Java. And this allows it to be both standalone and web-based. And I won't be talking too much about technicalities today, but if you are a Java developer, you can find out more about how Jalview works at jalview.org, and we'll be going there in a second. It's free software that's been developed by the Barton Group since the late 90s, in fact. So let's head over to Jalview, and I'll just show you a little bit about the website, because there's a wealth of information here. The most important thing is the fact that we have a news section. And we'll make announcements about new releases and also if there's anything happening about concerning our web servers. For instance, we actually our services went down for a couple of hours on Tuesday, so we put announcements up here. The other important place to go is the help section, and I'll just click to here so you can see. This lists the most important places to go and get help about Jalview. There is a comprehensive documentation page. There's an FAQ, which covers most things, I think. And also there's a Jalview discussion list. And that discussion list, it's an emailing list. You should sign up to use it. It's very low volume, so don't be afraid to sign up. Now I'll start the program and show you what happens when you run Jalview for the first time. So I'm going to run the latest development version of the current release branch of Jalview. So we maintain 2 or 3 different versions of Jalview. There's one which is probably the one that you use over at SB Grid, and we've got one that's very much a development version, and I wouldn't let anybody use that at the moment. And the version I'm going to run mostly is bug-free. I fixed a few today, as you'll see when it starts up. So there are a few warning dialog boxes that appear. In my introduction concerning Java, I used to say, well, it means you can run a program anywhere at any time. But recently it's becoming more interesting running Java, so we've got a few bugs to fix with that. Anyway, the first thing that pops up when you run Jalview, apart from the splash screen, is some information asking you things like whether you'd like to contribute usage statistics. Now, I very much like you to click yes, and I'll do this for this particular example. Normally I hit cancel or no here because as a developer I shouldn't be bumping up our statistics. We use this to quote to our grant funders. So the next thing after this is the news dialog box, and this is basically the same as the website. You won't see this dialog box unless something new appears on the website. So normally once you close it, it just disappears. And what's left is the Jalview example alignment. And this really demonstrates everything about Jalview. There's a structure window, there's a tree window, and then there's the alignment window, which is the business end of Jalview, really. There's also some toolbars right at the top on the desktop area. And on Windows, you'll see these attached to the Windows, to the desktop window, rather than sitting in the menu bar at the top on a Mac. The alignment window itself is in the desktop in the example alignment has several views created on the alignment, and if I press X, this actually expands all these views so that you can see them separately in separate windows. And you can see that actually if I do an edit in one of these windows, everything changes in the other windows as well. So this is the same data visualized in different ways. And the idea of this is that it allows you to actually prepare either different panels of figures, because each one of these has completely independent font sizes. You can increase the size or change the font. You can also hide and show different types of information. So these little blue triangles are highlighting the fact that there are some hidden sequences there, and I could reveal them here. And you can get an overview for each window as well, which just gives you the context compared to the whole alignment. So in this display here, you can see that I can move around. The red box shows the area that's in the current display, and the dark grey bars are hidden regions which aren't shown in this particular view. So if I just gather these together again, I'll press G and they appear all here. You can rename these, of course, so that you can actually give them some informative names. The tree viewer is actually linked to this view that I'm browsing at the moment, and you can see if I click on this tree was actually calculated in Jalview using the calculate tree option, which has been there for forever. You can also load trees onto Jalview and it will match them up with the sequences and allow you to do the same thing. You can use the tree to select sequences in the view. Clicking on the tree subdivides the alignment into groups, and each group of sequences it can be selected as a unit. And you can also see under here there are annotation tracks which show the consensus sequence for each subgroup, and also a kind of sequence logo. It's not an official sequence logo, but it gives you an idea of the distribution of residues in each column. And this allows you to summarize a larger alignment and to really navigate around with subgroups. And if you double click on one of these annotation tracks, you can select the group that it's associated with. So now onto the structure display. So the structure display is given by JMOL, and you'll notice that this structure is nicely colored but not at all associated with the coloring in the alignment view at the moment. That's because it's colored according to a different alignment view. This one shows various sequence database annotation highlighted on the positions in the sequence. And some of this is uniprot annotation, and I can hide and show that annotation using this view settings option here. And I can get access to the feature settings and control it in detail here. So I can enable and disable, for instance, the annotation that Jalview puts onto the sequence that highlights the residue numbers in the PDB file that have been associated with this spinach ferredoxin sequence. The colors can be configured independently of alignment view as well, and positions in the structure get highlighted in the sequence, so you can see that, and the same the other way, so you can locate yourself. And the structure viewer can also cope with more than one structure at a time, but we'll come back to that shortly. So the last thing I'd like to show you is the preferences options in Jalview's desktop. Now the most important thing here is this little tick box next to the open file menu. And if you untick this, that allows you to disable the display of the example file on startup. You wouldn't believe how many people who I've gone along to help who've been using Jalview for several years, and their first 30 seconds of Jalview use is to play catch the window and close every window that appears automatically. So just untick this box if you find yourself doing that, and you'll not see the example alignment again. Also see there's lots of default settings that you can change here. So I do advise you to take a look and set things up exactly as you like it, and if there's a preference missing, we can add it in. Okay, so I'll just close up all the windows there and head back over to the presentation. So typically, people use Jalview in kind of the following sequence. Firstly, they get the data into Jalview somehow, and you can drag sequences onto Jalview with various flat file formats. It can load from URLs and so on. You can copy and paste in as well. You'll then use the database retrieval functionalities to grab any database cross-references and sequence annotation that might be available on your sequence. And of course, you'll create alignments, and we'll do a little bit of that. Finally, you'll be using the built-in shading, the conservation analysis, the tree, and the PCA capabilities to explore the alignment, maybe refine it, and use any structure data if you have that around. and of course you can select regions, you can refine them, you can interactively edit, or you could use the alignment functionality again to refine sections. And maybe if you use other programs, you'll paste that data out into some other program and then import the result files back into Jalview. And finally, of course, you'll prepare your figures for publication, which was the original idea for Jalview adhering. And it's these highlighted steps are the things which the example file really demonstrates. It shows some examples of an in-progress analysis with some pre-prepared figures as views on the alignment. And I've already told you, you can disable the start of the example project in preferences. And the other thing that the example project demonstrates to you is what kind of information gets stored when you save a Jalview session. So Jalview projects are things which allow you to store everything about your analysis at a particular point. They save the alignments, they save the various views on the alignments, all of the annotation and database cross-references. If you've loaded PDB files onto sequences, then it will store the PDB file in the session as well. And of course the trees and display settings as well will be saved. And these are shareable. We spend a lot of time making sure that when we release a new version of Jalview, it can load all of the old project files, because we wouldn't like you not to be able to access your data. So I'll just go through these first steps again, and I'll drag a sequence onto Jalview and do a database retrieval just to show you how that looks. So my sequence is in a file sitting on my desktop, so I'll drag that back over to Jalview. Oh, that was the wrong way. Go this way instead. So that just loads in, as you might expect. And you can see that there are Uniprot identifiers here, and I can ask Jalview to use those to look up Uniprot records in the standard databases. Now, I've got the Jalview console window open so you can see stuff is going on. But it's very quick, and as a result, you can see there's lots and lots of information that's been mapped onto these sequences. This is interleukin 7, so it's the growth factor that has some interesting properties, which is why I've kind of shown it. And you can see that there's quite a bit of database references, database annotation there already from the Uniprot record. There are structures for this as well. And if I hide off the chain annotation, you can see most of what's going on here. And I'll just make this in wrap mode so you can also see there. So one of the interesting things here is there is the splice variant, and and that's just in the human form. There's nothing really characterized in anything else. And the human is the one which has most information about it. There you see there are 3 PDB files. Now I'm just going to actually disable the database cross-reference information in the tooltips, because there are so many that it gets in the way. Okay, so we have our sequences in Jalview. I can run an alignment using the web services submenu, and there's an alignment menu in here. And I'll leave this going whilst I talk a bit about the alignment services that Jalview provides. And I'll run a ProgCons alignment, and see that just sits there. This is actually running in Dundee at the moment. You can see the references for the program there. Okay, so sequences. There are a few different ways you can grab database annotation with Jalview. One is to just paste in a bunch of IDs into the sequence fetcher window, and you can pick from one or many databases to query with this. That's the way that some people work. Other times you may well have alignments already prepared somewhere, or just flat files with sequences in them, and you want to grab all the database references. And that's where this fetch database reference entry in the web services menu comes in. This is really useful when you've got fragments or subsequences from particular records. So say you've only expressed one domain in a particular gene. You need to get the numbering right in order to display the annotation in the right place. And Jalview will use the database ID to look up the record and then find the location of your sequence in the alignment in the in the official record for that sequence, and then adjust the numbering appropriately. This is something that I pretty much came to work on Jalview in order to do. I was looking for a program to do this 10 years ago, and there wasn't one around. And it's still tricky to do it these days. Of course, the point of this is so that you can get annotation in the right place, so you can use it to curate your alignment and also to pick up any PDB structures associated with your sequences. There's quite a lot of information presented in all the various tooltips. There are URL links which you can grab. If you right-click on a sequence feature highlighted on the alignment which has got one of these link symbols, then you'll get a list of URLs that you can browse to as well. And if you hide off or disable the display of the tooltip, you can also get access to all of the sequence annotation information in the sequence details submenu, which opens when you right-click on a sequence. And I'll just do that now just to show you. So this is the alignment that's come back. You can see there's the sequence details there, so I can get a dump of all of the database information there. Minor bug in Jalview. It doesn't inherit some of these settings, so as it ought to. These are preferences that you can set, but I haven't set them in this version. Okay, so back to the plot again. So there are various sources of sequence feature data. Jalview talks to the distributed annotation system, as well as the hard-coded databases that we serve from Europe. The distributed annotation system has got about 300 database sources, sequence data sources, and almost 1,000 sources of annotation on sequences. Some of those are very specific databases, which are not generally useful, but some are quite useful. You can also prepare or import Jalview sequence feature files and GFF files. GFF files are reasonably widely used. Just tab-separated files identifying start and end numbering and the type of sequence annotation. Those are actually really useful if you have a favorite set of sequences that you always want to mark annotation on. So you can mark up a sequence in Jalview, you can create these sequence features interactively, and then you can save a feature file or a GFF file and then drag that onto any other alignment you're working with to quickly highlight your regions of interest. You'll also see annotation coming from various alignment formats. Stockholm and AMSA are the two formats that I know about, which have reasonably rich sequence annotation support. And we also get annotation from analysis web services. So you saw me creating an alignment. This is, of course, the most important thing that Jalview can do, although one would argue that curating the alignment is equally as important. The alignment's coming from a system called Jabbers, which is the Java Bioinformatics Analysis web services system, and we have a public Java server in Dundee, and it's maintained by Sasha Shersnev. It's also very easy to set up your own server, and you might want to do that if you want to run really big jobs, or you just want to you don't want to share your data over the internet. It's not actually insecure. It's not secure. And certainly, if you're worried about patents, you might want to set up your own server so you don't send your data outside of in-house networks. The algorithms that Jalview provides are as follows, and I'll just give you a little bit of a summary. I'm sure everybody's familiar with the idea of getting an alignment out of a BLAST result set. And when you take a query sequence and you search against the database, you get a set of hits. And if you take those set of pairwise alignment hits and turn them into a multiple alignment, so sometimes you call it the flattened sequence view, or the flattened hit view for a set of BLAST results. What you get is a multiple alignment where the mapping is optimized between the query and each hit sequence, but the alignment between individual hit sequences won't be optimal. Now, in order to optimize all pairs of relationships in a multiple alignment is hard computationally. So various heuristics that were developed. The most popular one is the one used by Clustable W, and this is progressive multiple alignment, where a kind of approximate phylogenetic tree is constructed, and that's used to scaffold the optimization of the pairwise alignments between individual sequences or groups of sequences. The other extreme is something that's provided by T-Coffee and Probcons, and that's a transitive multiple sequence alignment. And here you use some Monte Carlo or other kinds of optimization to actually iteratively refine and improve the multiple alignment by optimizing all pairs. And there are various tricks to get that to run quickly. Muscle and Math are programs which provide hybrids where they employ profile alignment methods, which also allow some kinds of transitive alignment as well. Typically the problem is you have to rebuild the guide tree, so there are different ways that you can recalculate guide trees and rescaffold your alignment. The most recent alignment program I know about is Cluster Omega, and that's provided as the most recent service added to Jalview. And this can align startling numbers of sequences, and it uses some clever sampling to allow you to create very big alignments in a quite short space of time. And if you believe S. Higgins, then this produces almost as good alignments as T-Coffee. Okay, so the way Jalview provides access to alignments is through the alignment submenu in the web services menu. In addition to running with default settings, you can run with a range of presets which are provided by Jabber servers, and these might be quick settings, so you can put in quite a few sets of sequences and quickly get a result, or they might be much more accurate settings. You can also create your own settings, so if you go to the Edit Settings menu, you can tweak the command line parameters, and you can save those along with your Jalview profile. And you can also share those with other people if you want. So once you've got your alignment in and you've started to explore it, you might also want to run some other kinds of analysis programs. And again, one of the earliest analysis programs that came into Jalview was the JPRED system, and this allows you to perform an analysis of your sequence based on a sequence profile to predict its secondary structure, and it's simple three-state prediction, so helix, sheets, or coils, and it uses a sliding window to predict this. And there's also a coil-coil predictor and a surface burial or surface accessibility predictor. You can send it a single sequence, and it will do a side blast run behind the scenes, and then to build a profile out of the hits. Alternately, you can send it an alignment, and it will just directly generate profiles to do predictions on. And what you get back is something that looks kind of like an alignment, except it isn't quite an alignment, it's actually a profile. So the top row of the result is the sequence that was used for prediction. The rows below are actually the sequences that contributed to the sequence profile that was used for the prediction. And then finally you've got these annotation tracks right at the bottom, which are, starting from the bottom, you've got the JPRED line, which is the final JPRED prediction for the secondary structure for this sequence, and then the raw predictor outputs, JNet align and JNet HMM, and then after that there's solvent accessibility as well. So the more B's, the more buried your region is, that particular region is in the prediction. The next bunch of predictors you might want to run is the protein disorder predictions, and those are what we're going to demonstrate with these interleukin sequences. So I'm not sure how many of you are familiar with protein disorder prediction. I'd imagine if you are structural biologists, you probably have started to get quite concerned with that. It's a complementary problem to predicting structure, because the idea is to recognize regions which may not adopt a stable structure or may adopt multiple different structures. You can think of it quite trivially as actually learning to predict which regions of a protein are never going to be visible in electron density. So you think of it like you're predicting the REM 465 remarks that you might see in PDB files. Alternatively, it's predicting loops which might be variable, so that's the hot loops predictor. And you actually see this as annotation in Jalview when you get the results of these things. And then also you see domain annotation. So many of the disorder predictors take a low-level per residue prediction, and then they analyze the distribution of disorder in order to identify regions which might fold up into a globular protein versus regions that are simply going to be unstructured, and they might be linker regions, or they might actually form whole unstructured domains, which you wouldn't see otherwise, and would ideally cleave out if you were wanting to do a crystallization experiment. The methods that Jalview can access is a machine learning method. The Dissemble program provides this. There's the method called RON, and this uses a system where it has a database of known disordered sequences, and it measures the similarity between your sequence and that data set. So it's kind of an expert system in some ways. And globplot and IUPRED are empirical methods which have got a statistical model of disorder, and they as a result they're very quick, and those are the ones I'll demonstrate at the moment. Okay, so let's go back to Jalview and run some disorder predictions. So first one I'll run is globplot, and what comes back is some sequence annotation. So here there's a globular domain annotation, and also some local disorder predictions here. So these correspond to the regions which are scoring above the significance threshold for the globplot predictor, and you can actually see the raw output for globplot here. It's shown as an annotation track below the alignment. Now, if I look across here, there's this region where there's a reasonably reliable, or at least a reasonably conserved region of disorder, and then this earlier region I was looking at doesn't look so conserved. So we might want to take a look at that with another predictor just to see whether it's actually reliable or not. There's also this other disordered region here. So let me run another predictor, and I'll run IUPRED this time. And I could edit the settings, but I'll just run the defaults, which actually just does all the predictions you might want. And this again produces globular domain annotation, and here actually all of the sequences end up being predicted as globular, which is good. But I'll hide those off. And there's no other protein disorder prediction information shown on the sequence apart from this globular domain information. IUPRED also produces annotation tracks, and it has two predictors. It has a short predictor and a long disorder predictor, and those are parameterized for predicting short stretches like unstructured loops, or longer stretches like unstructured linker regions and domains. And you can see again there are threshold lines here which have been shown on the annotation track. And I'll use Jalview's color by annotation function to map those onto the alignment so you can visualize them more effectively. Now, out of the box, Jalview allows you to map any of the annotation tracks as shading on the alignment. And I'll just pick the per sequence annotation, and in which this case we've either got globplot or the IUPRED predictors. And I'll take a look at the short predictors, and then use the threshold setting to highlight just those regions which are actually above the threshold. And I've ticked the threshold as min-max, which means that yellow regions are just borderline cases near the threshold, and red regions are the most likely disordered according to IUPRED. And you can see here that the region in globplot that was not particularly conserved is again not particularly conserved in IUPRED, and in fact in the long disordered regions it's barely borderline. It probably is a false positive there, whereas this region is most definitely coming up as disordered. And of course, because we've got structural data, we can also pop those into a JMOL view and take a look and see where they actually sit in the structure. And so this is just a kind of a canned visualization that Jalview provides. If you've got structures on the sequences, you can select all the structures and then open them all in one and Jalview will then try and superimpose them according to the alignment. And you can see here that there's this nice long loop region on the outside, which is coming out as quite unstructured, and then everything else is looking as structured. That sort of false positive hit actually is corresponding to if we go a little bit further back. So it's actually this region that's poking out here, which doesn't look like it's forming a perfect helix. It might be another conformation though. Of course, that's actually a model, I think, in this case. Okay, so we've reached our half an hour, so I should wrap up. So Jalview provides one alignment and many views onto particular alignments. You can copy paste in and out and so on. You can also map sequence features to allow you to curate and inspect your alignment further and map more information onto it. And annotation tracks below the alignment give you information about the alignment quality, and also allow you to create your own annotation marking important columns in the alignment. There's the tree viewer, which allows you to quickly drill down and analyze the variation and subgrouping in the alignment, and the structure viewer that allows you to look at the structural context of your sequence alignment data. And this group selections from the tree viewer allows you to analyze subfamily variation, and colors and mouse overs can be transferred from alignment views and actually into the structure display. There are other structure visualization tools in Jalview. I've not talked about the RNA stuff that's appeared in Jalview in the last release. We in addition to JMOL, which is one GPL structure visualization tool, we've also got Varna, which allows you to view RNA 2D structure diagrams in Jalview, and you can actually do that interactively as well. So you can use the 2D view to navigate your alignment and highlight stems and helices. Generally, Jalview is an interactive editing and visualization tool. It's got these built-in consensus conservation and clustering algorithms and pairwise alignment. It's got figure generation capabilities. It can retrieve alignment data from PFAM and RFAM and FLUT files, various different ones. Retrieve PDB structures directly from the PDB in this case, and also from your own PDB files, so you can read and write those, in fact. Sequence data from Uniprot and European Nucleotide Archive, distributed annotation system, and also sequence annotation from Jalview feature files and GFF. Annotation can also be retrieved from Jalview annotation files, and of course the web services, and T-Coffee score files, which is a specific thing that some of you might know about. Last but not least, you can read and write Newick tree files. The suite of web services provided by Jabbers include multiple alignment and alignment conservation analysis, so that the AACon service actually provides 18 different alternate amino acid conservation measures that you can view in Jalview, and protein disorder predictors, which you've just seen. And of course, there's Jaap Heringer's group developed a system called multi-harmony, which allows you to analyze an alignment that's been subdivided using a tree to explore subfamily variation and key functional site variation, and of course spreading secondary structure prediction. And that's it. And I just thank the people involved. And,
Great. Thanks, Jim. If anyone has questions, feel free to put them in the chat window, pass them to me, and I will pass them on to Jim. I had a couple of questions. First, what are your sort of best sources for Jalview documentation? If somebody wants to, has a particular problem they want to address and they want to sort of dig into the documentation and find the best way to do that, what do you recommend?
So the first thing is to take a look at the interactive help. So this is searchable, and in fact it's online as well. So I can say if I want to work out how to look at the physicochemical property scores to find out how they're calculated. So the conservation coloring mechanism gets picked out immediately. I know this because there's very few pages on this site which actually use the word physicochemical. So there is an awful lot of information in the Jalview documentation. We try to keep this up to date and try to make it as informative as possible. But there's also on the website, if I have a look at the documentation page, You can also take a look at the Jalview manual, and this is 70-odd pages of exercises and explanatory text. And we actually use this for our Jalview courses, so this is kind of like the textbook that we base our training courses off. So you'll probably find pretty much everything there. If not, come along to the discussion forum and post an email there.
All right, great. I have another question came in. How do you associate structure from the PDB with a sequence in the alignment?
Aha, that's a good question. So, a couple of different ways of doing it. If you know the PDB ID and it's not already been recovered by doing a fetch database references routine, then you can just type it in directly. So that just allows you to associate any ID. What Jalview will do then when you ask to view the structure is it will import the PDB ID. I'm just thinking actually, if I try the 1IL7, this might or might not work. This is one of these crazy potential things. So once it's been typed in, it's just listed in the View Structure menu. And I'm going to open a new view, and you can see here what's happened is that Jalview has aligned the PDB sequence, all the chains in the PDB sequence in the PDB file to the given sequence that I was associating it with. So you can do the same thing just with a file that you have locally. So if you've got a new structure, you can just use from file to pick the structure. And now, many of you, I'm sure, work with sequences which are actually named according to the files in which your structures for the sequences are stored. And in that case, you can actually just drop the file onto the alignment, and Jalview will match up the named sequences with the named structures automatically. So if you've got a directory full of PDB files, and you always work with those sequences named in that way, and Jalview will be able to match them up automatically.
So I have a related question. Can you get the secondary structure info directly from the loaded PDB?
So in the current version of Jalview, and the one that we're going to release next week, it's not possible. This is actually possible in the development version of Jalview. I've been working on enabling this, and so JMOL actually has a built-in DSSP implementation, which we can use to extract secondary structure information. And so it'll get shown actually in pretty much the same way as the secondary structure information from JPROD. So you'll be able to directly compare the results of a JPROD prediction to PDB structure, secondary structure information. The problem at the moment is that when you're dealing with lots of annotation in the annotation window, it starts to become a bit difficult to manage all that information, and Jalview doesn't reorder the annotation tracks automatically. So I'm holding this feature from the current release because I'd like to do some testing with some people. So if you're interested, take a look at the development version, and I'll make an announcement on the Jalview discussion forum when we've got that feature ready for trial. And I'd really appreciate some feedback. So.
Great. I have one more question. Someone's wondering what you would suggest for a membrane protein for evaluating a membrane protein, particularly in regards to disorder, sort of, in sequence space, without regard to a lipid environment.
Yeah, I don't think any of the disorder predictors we have at the moment from Jabbas deal very well with membrane proteins. They tend to highlight the membrane regions as disordered, because of course they would be if they were in normal hydrophilic environments. I'm not sure what I'd recommend. I'd probably, and this is kind of drawing back from my old days as a protein structure predictor person. I would use a combination of methods. So I'd certainly use something like Phobius to do, or TMHMM to identify the likely membranous regions. And then in the case of the extracellular domains or cytoplasmic domains, I then look at disorder scores for those as well. I believe at the moment, I think is it Poodle 2? That's the protein disorder predictor that tends to be the best. But people are really actively working on this at the moment, so I'm probably not the best person to ask.
So one more question, and a sort of fitting way to wrap up the discussion here is, what's the best way to output your alignment for publication? Once you've got everything, which file format do you recommend?
I recommend that you should be using an EPS file, and if your journal does not take EPS, you should insist that they will. I think pretty much everyone does these days. So the EPS output that Jalview provides is WYSIWYG. It's using the same code to render, and I'll just create a file here. So test output, and when you export output in Jalview, you've got the option of leaving text as text in the EPS file, or you can render it as line art. Line art is the most accurate output, but the result of that is that if you want to edit the labels in a program like Illustrator or Inkscape, then you won't be able to do that by just double-clicking on the labels and just changing the text, because line art will render all the fonts into curves. That means that you don't lose the font if the people who receive your EPS file don't have the font on their system. But it does mean that it's more difficult to edit and tweak afterwards. So I guess does that answer your question, I hope?
Yeah, I think that's it. Great. So with that, I think we'll wrap up. Thank you very much, Jim, for a great webinar. And yeah, great. Thanks. And thank you, everybody, for joining. See you in September.
Mm-hmm.